First off, let me first introduce myself. My name is Brooks Bonner. I am the Director of Community Engagement and Enrollment Management for the Organization for Tropical Studies. And we are proud to roll out this new initiative called TropTalks, which is our online science webinar series. This specifically is the category called Stories from the Field, where it's going to feature prominent OTS staff, faculty, recent alumni, who will talk about their recent research that's done oftentimes involving courses that we run, as well as research that's done and conducted in and around our field stations, both in Costa Rica and South Africa. So I'm super excited today to introduce Dr. Sofia Rodriguez-Brenes, who heads up our education efforts in Costa Rica. And she studied biology at the undergraduate level from the University of Costa Rica, and then earned a doctorate in ecology, evolution, and behavior from the University of Texas at Austin. And much of her research is focused on the Tundra frog, which we will be hearing about today. And I've had the pleasure with working with Sofia for the last year plus, and hopefully all of you are as excited as I am to hear and see this presentation today. So without further ado, I'll pass it off to Sofia to take the microphone and yeah, take it away.
Thank you. You can hear me, right?
Yes.
Okay. Thank you, Brooks. Brooks only gave me 15 minutes, so I'm really going to try to stay on time. So as Brooks said, I've been working with frogs, and today I'm going to talk about frogs in the age of extinction. So why am I saying the age of extinction?
Sofia, I don't think we can see your screen.
Really?
No.
Let's see. Are you seeing it now?
Yes.
There.
Okay, great.
So why am I saying the age of extinction? In the last decades, we've had plenty of evidence that lots of animals are going extinct, that species populations are declining. And a lot of people have been calling this age the sixth extinction. So the frogs have been doing pretty bad. Well, amphibians in general, about 1/3 of amphibian species right now are threatened. And a little bit less than 200 species have already gone extinct. So the causes for these are very diverse, and some of the usual suspects. We know of habitat loss, of course, can affect many groups of animals, pollution, climate change, some diseases, and of course, the combination and interactions among all of these. So I'm going to talk about one of these diseases that is called, or is caused by a fungi, a chytrid fungi called Batrachochytrium dendrobatidis, right? We're going to call it BD. It's a chytrid fungi, so sometimes I cannot avoid, but sometimes I refer to it as chytrid, sometimes as BD, but just know that I'm talking about the same thing. So these chytrid fungi causes a disease in amphibians called Ketomycositis. This fungi was described in 1989, so it's fairly recent, and it only affects and has been found to only affect amphibians species, and especially their skin. So we know that in frogs and salamanders, the skin is very important because they do most of their gas exchange through their skin. So they breathe through their skin. So it's key in their biology. So BD infects the keratinized tissue in adults and in tadpoles. In frogs, the keratinized tissue is their skin. So if you see here, this is if we cut the skin of a frog, you can see the dermis and you can see the epidermis, and this is where the tissue is keratinized. So these things here, this is the skin of a frog that is infected. And here you can see the zoosporangia, where the spores of the fungi is growing. And then when it matures, it bursts into the surface through some pores. This would be the surface of the skin, and it can infect the frog again, and it can infect other individuals. And why is it that affects them so much? It is because as this layer of skin gets infected, the chytrid is doing it's very happy over there, and the skin starts getting to thicken. And this thickening, it disrupts the function of the skin, so the frogs cannot absorb water, they cannot breathe anymore. And for some species, this disease is lethal. But for the other ones are not lethal. So we can find them in the field. They're infected with the chytrid, but they don't seem to be dying. So I was interested in understanding when the chytrid doesn't kill in these species, what was happening. So I studied the Túngara frog that is really common along the lowlands of Middle America. Here's the distribution of the Tundra frog. So this is Costa Rica and Panama, and these orange patches is where we can find it. And so it's important to say that they are distributed along the lowlands, because in the lowlands it's warmer. We know that the optimal growth temperature for the chytrid is between 17 and 25 Celsius, which is cooler temperatures that we find in the highlands. In the lowlands, we find species like the Tundra frog that we have found it infected, but the population seems to be doing fine. So it seems that it's not lethal for them. How do we study the fungi? So we go to the field, we collect the frogs, we swab the ventral skin where it happens to be more common to find the chytrid. Then we take that swab to the lab and we try to see if we find DNA of the chytrid, because we cannot see it, but we can tell if there's DNA, then that frog was infected. So when we started doing this, 2010, we started surveying some populations along Costa Rica and Panama. And there are lots of points here, but we're gonna focus on only 2 of them. So we had evidence that in around, for example, 95, in this area here in Chiriquí, there's evidence that the chytrid had been there at the time, and there's a population of Tundra frog there. So we know that this population had been in contact with the fungi for a while now. And there's some other populations like these 2 where we find the chytrid for the first time in 2011, 2007. And before that, we haven't found it yet. So we have 2 populations where we know that some of them have been with the chytrid for a long time. Some of them just had been in contact with the chytrid. So I did a collaboration with a group that were studying this one gene related with the immune system invertebrates. But they found this one gene that we're gonna call it P9, doesn't matter, but it's related to resistance to the chytrid. And they found this in another species of frog, another population. So and they found that these frogs, the ones that were related, that were tolerant to the infection, that they would be infected, but there were no lethal effects. They found that they had all of them had this gene. So we wanted to see if in those 2 populations that I mentioned, if we could find this gene. So we looked at those 2 that I told you, like those 3 populations, but that had a different history of contact with the chytrid. And so in this one here, that is what gives us more evidence. This is the population that had been in contact with the chytrid for much longer, so from 1995 at least. And so here we see that this black bar says that every individual that we found had this allele, that had this gene. So it's telling us that even if that population had not declined, for some reason it was beneficial for those frogs to have a gene that seemed to be related to resistant to the chytrid. So here we were starting to have some evidence that maybe something was happening. So again, I wanted to see what was happening with this species that we know that it was not being affected, or the population was not declining. So I was interested in the reproductive behavior. So I wanted to know if the infection with BD had an effect on the reproductive behavior of the Tundra frogs. And now I wanted to focus on 2 aspects of the reproductive behavior. The male's mating call. So the male frogs may produce a call that is to attract the females. So I wanted to know if the mating call characteristics would vary as a function of their infection status. And I wanted to know about the female choice. So if the females could discriminate against the call of those males when they were infected with BD. And as Brooke said, we cannot talk about the Tundra frog and their calls without hearing the Tundra frog. And so here you can be able to hear them. So they are nocturnal. They make choruses and they call from the water. And they sound like a video game. I'll stop it there. I can get tired of hearing them. And so how do we study the call of a male? So we go to the field, we record them, and then we can put those sounds in the computer and we can visualize the sound. How do we do that? So here is how the sound looks. So in the x-axis, you have the time. And in this graph here, what you can see, we see on the y-axis is here, the y-axis, you have frequency. So this is like low frequency. So they're like very, yeah, like low, very low frequency, like, and here we have higher frequency that are just sharper sounds. And then here in the x-axis, again, you have time. And here you have how loud does it sound that call. So at the beginning, you see that it's very loud and then it gets quiet and it gets very loud at the end. And we can measure lots of different things. So here I'm just showing some of the things that we can measure in the call. And then how do we ask the females what do they prefer? So we do a really simple test that is called a Phonotaxis test. And what we do is that we put the female in a sound chamber. So just like a small room where we can put in and it's soundproof. It's to a scale. So this tiny thing here, it's a female Túngara frog. And then in each side, we put a speaker. And from each speaker, we can play back different sounds. When the female starts hearing these sounds and she really likes one of those, if we put male calls, if she really prefers one of those, she moves toward that speaker, sits in the front, and doesn't move from there. So we can tell that she prefers what call she went towards. So what I did was playing the call of, let's say, male A that was infected with the chytrid and male A when he was not infected. What we do is that we treat them with a fungicide and we can clear off the infection. And see, we would see, we did this with several females and we'll see which one did she prefer. So after many, many tests, we did this with the calls of 3 males, say male A, male B, and male C. And here on the y-axis, we have the proportion of female choices. So how often did the females went to either of these calls? And here you can see that in blue is the times that the females preferred the call of the male when that male was not infected. And in red, the calls of that male when he was infected. So it's very clear that the females prefer the calls of the males when they were not infected. Now, there was a very obvious characteristic that we saw that vary between these two calls, infected versus not infected, and it was that the males, when they were infected, were calling quieter. And that's usually, it could be a sign that they're weaker. And so the females usually prefer sounds that are much louder. So we can manipulate the sound. So we did some manipulations now to make the frogs that were sick, to make them sound louder. So we would think that that's more attractive. But still, after doing all these manipulations and after testing many, many females, we could see that they still prefer the calls of the males when they were not infected. So there's something else there that we are not measuring. So after this test, we can tell that regarding the male mating call, the acoustic properties of the male's mating call are affected by the infection with BD. And regarding female choice, we can tell that the infection status does affect the female preference. So overall, now we know that infection with BD in tolerant species might have a cost on males. So it might be that the populations are not declining, that we don't see them dying, but if this is affecting a male and cannot successfully reproduce, cannot find a mate, then it can have long-term effects. And this was only one species looking at one disease, and we're only looking at one aspect of the reproductive behavior. Now we have to see what is it that is causing those effects. So it is very clear that to be able to have more informed conservation efforts, we have to have long-term studies. This study took, what I did in 15 minutes or less, took 6 years. So we really need to do long-term studies to understand the threats that are affecting populations in general. And at OTS, we welcome and support long-term research projects, but we also want to get the students more involved in these studies. And we want that other projects, researchers will come to the field stations and they can build upon these data. And we've had the history already of amazing long-term studies that have happened in our field stations and that are key in certain aspects that we can understand on climate change, for example. And we want all these efforts to that will lead towards more informed conservation actions. I don't want to miss thanking lots of people that I work with, and I'm forgetting lots of people, I bet, and institutions that help with this study. And with that, I did it in less than 15 minutes. I want to thank everybody. And then if anyone has questions, I'll be happy to answer them.
Thank you, Sophia. That was great.
You're welcome.
That was great. And I think I'll just say that, if people want to ask questions, they can unmute themselves and ask away. They can also put it in the chat if they want, if they're feeling shy. But I wanted to make the connection too with, I was mentioning earlier before the talk started that there's, an effort to kind of revamp and restart some of the undergraduate programs in Costa Rica, which Sophia is a huge part of, that we hope that students as they come down will develop many of these field research skills that Sophia has shown, and that they can take part in some of these long-term studies, because we know that we'll have students coming down at certain times of the year, which is a schedule and is a format that has worked well with our courses in South Africa as well. So we hope that students come down at the undergraduate level, but also continue at the graduate level as well. We just had our first kind of hybrid field course in the COVID-19 era, and it went off successfully, and with all the precautions that we took place, and it was part of Sophia's efforts to make sure that course went smoothly without any major problems. So I want to give you kudos there as well, Sophia.
Thank you.
But yeah, I want to just open up the floor to everyone to ask questions, and yeah, feel free to just speak up.
I was gonna say this is gonna sound like advertising, but every time I think of how I approach my research projects and questions, I go back to when I was a student in the graduate course, and you can really see how my research has been shaped, but by what I learned back in those times. So we'll just hope to keep giving those skills to students to come. Mm-hmm.
I had a couple of
Mm-hmm.
questions. So the first one is, do people know whether the fungus affects more males or females, or if there is a difference? Oh, and then the caption.
I don't think there is a difference. I haven't seen where if someone had like divided between, or like see if there's more females and males, and I don't think so. But for example, I didn't say this, but we tested the females as well. So when they will put a female in the chamber and see her preference, and it didn't affect the outcome. So it didn't matter if they were infected or not. It didn't matter which one would she prefer.
So then
Yeah.
my other question is, so for males, it seems that it obviously has a big effect because it affects their calls, which means that they're less successful. But like for the females, like what would be, like, would it affect their egg laying, or like the embryos, how successful those are?
I don't know, and something that I didn't talk here is that, so when I put the male and when I put a female in the chamber, it's a female that I grabbed and it was in amplexus, so it was mating. So I know that um, choice was related to reproduction, because if that female doesn't mate in that night,
Mhm.
it's gonna lose its eggs, let's say. And so then if you put them back, they continue their mating and they produce eggs. So I could collect the egg mass and then check if that egg mass would be have more tadpoles, or they were growing faster or less, depending if the female and the male had was infected. So what I found, and the results are kind of complicated, but something that a trend that was very clear was that there was a trade-off of them when the males were infected, they could have they would produce more eggs, but the tadpoles were smaller. So there was some interaction with the infection in the chytrid, and I didn't find that. I also looked for like how well the males were doing. Were they like skinny, like their condition, their body condition, and there wasn't anything clear. So it's not clear. A lot of people have asked me, like, does infection just affects how they produce the coal? But the chytrid is infecting or it's present in the skin. It's not anything in the mouth parts or like
Mhm.
the larynx. So it's not clear. And as I said, there's something that I'm not measuring that the females are paying attention to. And something like how loud it sounds is something that we know that is related with body condition. So if a male is not doing really great, it's kind of weak, it's going to produce quieter sounds.
Mhm.
So I don't know.
That makes sense.
So
Yep.
is BD present in La Selva, and is any study have been done on trying to see if our frogs are not calling or singing nice as yours?
Oh, are you asked me? I don't think so, but we will do that.
But BD is
I
present in La Selva, right? And
yes,
our frogs.
yeah. In Costa Rica, you can find it most everywhere. Yeah. So it's in the lowlands, so that's why it might be that it's not that abundant and might not be lethal for many species. But again, we have to see a lot on what other aspects might be affecting. So we know that the species in La Selva have been declining, but we don't know why, and it might be one of these long-term effects that we don't know.
Hi, Sophie. I'm so
Hi.
happy to see you. I have a
Happy to see you
question.
too. Yeah.
The female that you use in your research were infected or not?
Some of them were. At the end, we had about 160 something, and we tested if they were infected or not, and if that affected their their choice for their call, and it
Oh,
didn't.
all right.
No.
Sophia, do we know, or do you know where the BD comes from, or?
It's a long story. Has been, of course, there's been lots of studies to see where does it come, and the latest evidence is that might come from Asia, and there are several strains around the world now, several strains, but not all of them are so pathogenic, so they might not be that lethal, might not make them so sick. But what has been found is that it might be that the strain that is more lethal, it's a hybrid between two of those strains. And yeah, but this, as I said, it was only described in 1989. That's fairly recently. So there's been lots of time trying to find these. And before these were all hypotheses from everywhere, Africa first, but now the last evidence is Asia.
Okay, all right. And but there's no but it's naturally occurring. It's not something that comes from like a human created product or something like that, is kind of what I was getting at.
No, not like human created. Definitely not. It has been found there's some species in amphibian populations in Africa that are infected with chytrid, but they don't have any disease. So it might be that was a hybridization between two strains. So hybridization is just that two different strains start reproducing among each other, and this combination made them very lethal. But no, not
Okay.
human made, definitely not.
Thanks.
You're welcome.
Sophia, I had two questions. One is the male has a two-part call, and the female finds, I forget, it's the whine and the chuck. It's the chuck that's attractive, right? So
Is the number of chucks comparing against, yeah,
right?
so between two males.
So what is that? What the infection may have been influencing that then influenced female choice? That part of the call.
So as I said, it was, and this study was hard because, as you know, the Tundra frogs have these two parts, and it had that like the louder and then that chuck. So that's why we call the one and the chuck.
Yeah.
So if you put a male that has lots of chuck versus one that has one chuck, the one that with more chucks is more
right.
attractive. So when I do this comparison, I had many more other calls recorded, but when I do this comparison of the same male with the chytrid and with not, I needed the male to make the same structure. So
Oh,
if
right,
it make
right.
a one and a chalk, I needed that afterwards would
Mhm.
produce another one and a chalk. And I can separate them, but we don't know if when you put the chalk away of a coal that was produced with a chalk, that really affected the wine. So I wanted to be very like strict on how I would put these treatments to make sure that
Yeah.
I was really understanding. And
Mm.
so I had like one with a wine, another one with a wine and a chalk, and I think two ones a wine, one and one chalk. So I measured the wines only. So what
Oh,
I
okay.
checked was the wines, and
Yeah.
the two properties that I found were different between the same males. If you
Mhm.
all plot the two calls, the 6 calls of the 3 males with and without, the amplitude, so how loud that would sound, and the dominant frequency, which is the lowest frequency that sounded louder. Those were the two things that changed. So
I
that's
see.
why I manipulated the amplitude, because that's
Gotcha.
also amplitude. We know that it's very important in
Yeah.
female choice, and that really didn't affect. That was
Mm.
very surprising.
Yeah. Okay. Then I had another question, if that's okay. You had showed that the 3 populations that were in the presence of the chytrid was found at different times,
Mm-hmm.
and it was that one population that was known infection in 1995 at least,
At
and
least.
and 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 the allele that's associated with tolerance is almost fixed, right, at 100%. So could that be interpreted that the chytrid was driving the fixation of that allele because that made it tolerant, and that would have happened in 20 some years, whatever, do the math, whatever it is. Yeah.
Exactly. We don't
Yeah.
know. And again, this gene was just like something that they found in common in the MHC complex that was
Yeah.
in other species, other continent.
Oh, okay.
But we don't really understand what is it that this gene does.
Oh,
So
okay.
it's like, it's just related. So we cannot be certain like, oh, this is what this gene is doing.
So it's
But
correlative. Yeah. Right. Yeah.
exactly.
Yeah. Okay.
Yeah,
Yeah.
exactly.
Yeah.
Mm-hmm.
You said that this fungus was in a number of countries. Is it also a factor in North America?
Yeah, this fungus now is in every continent where there are frogs. It's everywhere now. Mm-hmm. I mean, amphibians. Every continent except Antarctica. Yeah.
Does it? I'm assuming that it has a higher mortality rate just depending on the species and things like that.
Yeah, exactly. Some species you find them. So the species that seem to be more affected and very rapidly are species that are close to the creeks, to streams. Maybe because the zoospores, the spores of these fungi disperses through water. And also higher in the mountains, probably because of the optimal growth temperature for the chytrid. And some species that they seem to be doing fine. And they, in some places where you see like sharp declines, then you have some other ones that become super dominant very rapidly because they have like no competition. And yeah.
That's interesting.
Do they know if it can be transmitted from mothers to the eggs?
So the thing is that in most species of frogs, there's no parental care. So usually the frogs go and deposit the egg somewhere, and then they just might be where other adults are. So it's really hard to tell. But so something I didn't say is that the keratinized tissue in frogs, they have it in the adults, so it's in their skin. But the tadpoles, the larvae, don't have keratinized tissue in their skin, only in their mouth parts. So it affects their mouth parts, and when they're tadpoles, the ones that are really affected, they cannot eat because they mouth parts get all weak. But once they're going through metamorphosis, then their skin starts getting keratinized and they get infected. But it could be from the mother or just any other individual that was in the same, that just dropped some spores in the water and then it gets infected. Yeah.
Sophia, what precautions do amphibian researchers need to take?
Yeah. So, for example, at that time when we're going through all the population, seeing how often we'll find it, every time we'll go from one place to the other one, we'll stop at a car wash the car, bleach everything, bleach boots, because there was at when these started happening and there was like an alarm because some populations were doing fine. And suddenly we will see them declining from one year to the other one. The populations will go down really quick. There was a lot of research on trying to see how would kill the frog, and were we dispersing the chytrid, right? They did find it that it could stay for a little while in mud, so that's why everybody was bleaching their boots. But if it would dry out, then it would die. So basically, one thing that is very important, and I think we should be very aware of this now, is that it's very important that we're not just grabbing any animals, because even if we will hold a frog and then we go and hold another one, we might be passing some diseases that we don't know through our hands. So that's why, again, we have to be super respectful with diversity out there. And yeah, just be very aware that we might be passing things. And so as we're going from one place to the other one, wash equipment, wash your boots, and don't bring stuff from one place to the other one. It was a lot of discussion around that at the time.
Yeah, that's becoming more and more important these days with the
Exactly.
Yeah.
I could just quickly echo up here in cold Maine. We're right next to a national park and doing research on amphibians in the park. It is the guidelines are really strict about bleaching and wearing gloves and changing gloves every time you handle a new animal. So it's great that it's there, but it is really strict. It's for good reason. Yeah.
Exactly.
Yeah.
Sophia,
Yeah. See.
I don't know how many exotic frogs are in Costa Rica. I know Coqui is in Turrialba. Are those non-native species being affected by BD?
Good question. Ranacates bayana has been found as well, but I don't know. So coqui is something that is common in the lowlands. And again, lowlands are not so affected like lethally by the chytrid, but I don't know how they're doing. Good question.
Okay, and Coqui likes staying along rivers and streams. It's really
exactly.
localized in Turrialba city, and it's one of the main populations. I was wondering about that. Thanks.
Yeah, I don't know. Good question.
Good questions, everyone. If you got some more, please, I don't see any in the chat, but feel free, now's your chance. You may never get a chance again to get a question, BD. Okay, well, if there's nothing else, anything else, Sophia, that you want to share?
No.
Okay.
Hopefully the pandemic will be over and we'll see everybody in our field stations soon.
Very soon. I'll be there too.
Yes. Yes.
Gracias.
A ustedes.
Great. No, thank you, Sophia, for doing this. Appreciate it. Thank you for everyone that's joined us. We had a good audience here today. Great questions. And we will be doing this monthly, these stories from the field. So our next talk is going to be in exactly 1 month from now. And it's one of our guest lecturers in our South Africa office, who's going to be talking about a human parasite in birds in and around Kruger National Park. So stay tuned for that. If you signed up for Staying in the Loop with TropTalks, we will send out another announcement about that. But you can go to our website, tropicalstudies.org. Under the education tab, there's a whole page dedicated just to TropTalks. So feel free to sign up there. And also, if you're an alum or you know an alum that maybe is has done research on a cool topic that would love to give a short presentation like this, we are open to having other presenters from within the OTS community and network give other talks. So feel free to spread the word, and hopefully we'll see you all here in a month.
I'll be here.
Great. All right. Thanks, everyone.
Thank you, everybody.
Thanks, everyone. Thank you, Sophia.
Thank you.
Thank you.
Have a good night.
Thank you.
Pura vida.
Bye-bye.